Well, Happy New Year to you all. Welcome to our Tuesday seminars on the cerebellum. Today we have as our speaker Mohammed Amin Fakharian. Amin received his bachelor's and master's in electrical engineering from Sharif University in Iran, graduating with high honors. During his master's, he was mentored by one of my former students, Ali Ghazizadeh, who was studying brain networks involved in value-based decision-making and perceptual memory in non-human primates. Amin is now a graduate student in the PhD program here at Johns Hopkins, studying the cerebellum in marmosets. And he's been instrumental in describing this theoretical idea regarding how to put together cells into populations and describing this notion of a vector calculus in the cerebellum. So I'm happy that we get to hear from him and then here to listen from the feedback that all of you might have for this work. So, I mean, it's all yours.
Thank you, Reza. Happy New Year to everybody. So today I'm very excited to share our finding on population activity of different cell types in cerebellar cortex with you. And the title of our work is A Vector Calculus for Neural Computation in the Cerebellum. So cerebellum is essential for endpoint accuracy of the movement. And one of the common symptoms of cerebellar patients is dysmetric movements with the most obvious errors at the end of the movement. Here on the left, you see an ataxia patient who is not having that much difficulty starting and initiating the movement, but as you can see, it's hard to end the movement accurately and struggling. And on the bottom, I have the plot from work of Maurice Smith in lab, who had reaching a study on the cerebellar degeneration patients. And as you can see, the movements are initiated. They roughly have correct directions, but their endpoints are not as accurate as the control subjects. And on the right, we have another cerebellar ataxia patient, and you see the eye movements. And as you can see on the bottom plot, the eye movements in these patients are not accurate and they overshoot. So very similar to reaching. The movement initiates, but doesn't land correctly on the visual target that is present. And it fluctuates and fluctuates up until it eventually converges. So how does cerebellum do that? Cerebellar output, specifically the Purkinje cell responses, are heterogeneous, and this has been shown in multiple species and multiple tasks. This is reaching task in mice, and as you can see, there are Purkinje cells which increase their activity or burst, and there are Purkinje cells that pause or decrease their activity during the movement, and there are mixed cells. In addition to that, this has been seen for the whisker control in other parts of the cerebellar cortex for the Purkinje cells. This has been observed in the licking movements. As you can see, some of them are bursting, a group of them are pausing to the onset of the lick movements, and also in saccades. So we have bursters, we have pausers, we have burst pausers, pause bursters. And as you can see here, for the saccadic eye movements that their durations is around 50 to 60 milliseconds, the modulations are way beyond the movement duration. So how can cerebellum be involved in endpoint accuracy while the discharges of neurons are way longer than the movement itself? So to study the cerebellum, we use marmosets as a sensory motor model. They are primates and they have very rich behaviors. And as you can see here on the left, Charlie, one of our animals, is making goal-directed gaze movements, a combination of head and eye. And on the bottom, we have a goal-directed lick movement in our animal for harvesting food. Thanks to their size, in a head-fixed setup, we're able to record from thousands of neurons over a course of months. And for each neuron, we're able to record from thousands of trials, as you can see in the second plot. So number of saccades at the end of the day is going to be around 700,000 for 50 sessions of recording. And at the same time, we record licks. And in each session, we can record from, again, thousands of licks. So thanks to the silicon probes, we're able to record from 20 to 350 simultaneous neurons from multiple folia with up to 10,000 of licks and saccades per day. So the task that we have for the saccadic eye movement is that the monkey is head fixed and we present visual targets. We start with a fixation point. The monkey is required to fixate for 100 milliseconds. Then we show a primary target. And the monkey makes the primary saccade, and we jump the target when the saccade initiates to induce an error to one of the 8 random directions. So the task would include 3 main saccades, center saccades followed by fixation, primary saccade followed by the primary target presentation, and the target jump that induces a secondary saccade. And we detect the targeted saccades. We call them reward relevant saccades because the animal needs to make accurate movement to get rewarded in the task. And we also detect the spontaneous saccades that the animal makes out of the context of the task. So in each day, we are able to, using modified deep neural networks developed by Ziad Hafed Lab, you and I, we're able to record 10 to 20,000 saccades and microsaccades per session for these animals. Our recording setup, in order to be able to use the silicon probes, especially neuropixels, 10 millimeter neuropixels in these animals, we plan our craniotomies with individual animals' MRI and CT scans, and we plan our burr holes posterior. That's the probe trajectory would be around 4 millimeter, which allows us to put a 10 millimeter probe in. In addition to that, the area of recording that we're interested in, the posterior vermis, lobule 7, we're able to stay in that for the whole length of the probe. So a neuropixel probe with a 4 millimeter span is able to record simultaneously from multiple folia. And as many of the audience know here, the cerebral cortex has a 3-layer structure: molecular layer, Purkinje layer, which has the Purkinje cells, which are the output neurons, and the granular layer with plenty of granule cells in that area. So we have electrophysiology, and we have multi-contact probes. And as previous studies have shown, complex spike waveforms indicate layered structure. So using the climbing fiber signature, we're able to tell if we're in the dendritic tree, which the spike has a downward slow waveform, or near the soma, which has a sodium spike, followed by a calcium phase and the spikelets. Here is an example recording using neuropixels. And as you can see, the downward spikes with high rate are the simplest spikes. And the upward spikes that are happening around 1 Hz are the complex spikes. So here is an example recording. We had 5 layers of Purkinje cells, and these are cell types by layer identified. And here are the example waveforms. So here I have the first Purkinje layer. The dendritic tree is up because the climbing fiber activity has a downward here, and it has the somatic spike down, and the simplest spikes. And we have molecular layer interneuron, the dendrites of the Purkinje cell, and we have the Mossy fibers in the granule. And another layer, and the rest of the layers. And as the anatomy dictates, the waveforms flip when you go to the next layer because the folias fold over each other. So, is there a question?
Did it already? Yeah. Wait.
Excuse me? I cannot hear you well.
There was no question. Please go ahead and continue.
Oh, okay. So let's look at the modulation of the Purkinje cell simplest spikes, which is basically the output. And as we discussed, we are able to replicate the results that they were already explained, is that we look at the saccades to the other to different directions. And here I'm showing you example cells. So some of the Purkinje cells pause, some of them burst. So each row basically is one Purkinje cell, and these are the responses to different directions of the movement. So first thing is that they burst or pause toward all directions, and also their modulation lasts way longer than the movement duration. However, the simulation studies have shown that Purkinje cells pull the eye toward a specific direction, which we call it potent axis, which has been shown by Noda and Fujikawa by electrical stimulation, and more recently using optogenetic stimulation in macaques using by Shadmehr. So the question is that how are P cells involved in endpoint control despite the fact that they are active long after the movement ends? People have talked about this in motor cortex, and one of the more working hypotheses from the Mark Churchland lab is the null space theory, which suggests that the activity of neurons cancel each other during their relevant period. So the task that they have is that the macaque is sitting, and they basically require to do a reach task. And in the reaching task, there's a preparatory period that the animal is just shown with a maze that they're supposed to reach inside, but no movement is occurring. But what they observe is that the motor cortex neurons are active during the preparatory activity as well as the execution. However, the biceps, the EMG, is not active. So the question is that how come that the neurons are not exciting the muscles during the preparatory phase, although they're active? And the basic idea is that they linearly cancel each other in the null space, and the potent space would be where the activity forms the activity of the muscles. In order to model that, they use a linear model, and they basically linearly map the muscle space, which is the reduced dimension EMG that they record. The neural space, which is the reduced dimension rays, to the muscle space, which is the reduced dimension EMGs. However, as any other fitting, it has problems and difficulties. So the problem is to find a linear map between the neural activity and behavior. And the challenges are, first, there might be sampling bias in the neural population, which can be cell types and the location of the recording. Two, it can be choosing the behavior space that is relevant for the regression. And the third one is the uniqueness of the fit, that they try to use rich regression, but basically you need to put some assumptions to be able to have a unique fit to the data. The question that we are trying to answer here is that can we find a ground truth weight matrix or not? Actually, in the cerebellar cortex, we are blessed to have the climbing fibers and complex spikes, which have been shown us that it might be possible to estimate the weights without fitting a regression. So this is a recording from a Purkinje cell, and as you can see, Purkinje cell is generating the simplest spike and complex spikes. And usually a pause in the simplest spike is followed by the complex spike. And that is how we detect the Purkinje cells. We know that there are simplest spikes that have baseline around 50 to 60 hertz. We have complex spikes that have around 1 hertz of baseline activity. And the simplest spikes are followed by a complete pause after a complex spike. There's a pause in the simplest spike right after a complex spike happens for 10 to 30 minutes. This has been studied widely since 1992. Another property of the complex spike is that they induce LTP and LTD in the simplest spikes after they're followed by a specific delay after the parallel fiber. So here is the work of Jennifer Raymond lab. And as you can see, if after a parallel fiber, after a specific delay, a climbing fiber happens, the simplest spike activity changes, which is as a consequence of changes in the parallel fiber to the Purkinje cell synapses. But how do the complex spikes are modulated in the oculomotor vermis? So here's an example complex spike. And this is the response of the complex spike for leftward saccades and rightward saccades. As you can see here, this cell has a preference for leftward saccades. Before the movement, it increases its activity. And to the opposite direction, actually, it decreases its activity. And for the perpendicular direction, for the up and down, it's almost around the baseline. So we can present this, and all of the complex spikes almost in the oculomotor vermis have a directional response. We can represent this with a vector. The vector is going to have an amplitude, rho, and it's going to have an angle, theta. The theta is the preferred direction of the cell. We call it CS on. And the amplitude is the sharpness of tuning, how modulated this cell is. the cell not only responds to the movement, but also to the visual stimulus. So here is a line to the target onset. Here is a line to saccade onset. And as you can see, there's a peak associated with the visual. There's a peak associated with the movement, which can be seen when you align to visual. And as we go, you can see the tuning for different directions. So that was aligned to the CS on direction. This is 45 degrees off the CS on. This is 90 degrees off, which, as you can see, we are around the baseline. And as we go to the opposite direction, the activity is actually a decrease. And if we bin the responses by the amplitude rho, you're going to see that some of the cells are very sharply tuned. Here's the response for the CS on. Here's the response for the CS off. And here is for the CS plus and minus 90 that are at the baseline. And for some neurons, it's less sharp. They're less modulated by the movements and the visual targets. But in all cases, the complex spike is blind to the errors or the visual targets that are in the CS plus minus 90 degrees. Okay, so how can we use the complex spike to infer the weights? So there are two important studies here that the lab has conducted before. So let's say that we have a Purkinje cell, and we're interested to see, and from anatomical studies, we know that there are projections to the burst generators. And the motor neurons in the oculomotor plant basically have vertical and horizontal burst generators, which control the eye for upward saccades and horizontal saccades. And the diagonal saccade would be the combination of the two. So let's say that the Purkinje cells project to each of these with weights Wx and Wy. And the climbing fiber that is coming to this specific Purkinje cell has the CS on with angle theta CS. So what we can do is that the work of Solomon Muller is that complex spike can suppress the simplest spike during the saccade, meaning that the biophysical property that the simplest spikes are followed by a pause after, or having a pause after a complex spike can occur during the movement itself, which can be seen as a kind of perturbation to the normal activity of the simplest spike. So this simplest spike on average is responding as a burst during the saccade, but if a complex spike happens in this period, the burst does not happen and we have a pause. So what would be the effect of that on the behavior? And the effect is seen to be pushing the eye ever so slightly toward the CS on direction with no component in the perpendicular direction. So you can see for this diagonal saccade, if a complex spike happens during the movement, the movement is pushed toward the CS on direction ever so slightly. Another approach to see the same effect is through the CS-induced plasticity. So David Hersfield's work has shown that if in the previous trial, the error is followed by a complex spike, in the next trial, the simplest spike is going to have less activity than if the complex spike was not present. So a pause that is not directly because of the complex spike, but is because of the LTD that is happened in the previous trial. And the consequence of that, again, is shown to be very similar with changes in velocity in the CS on direction with no change in the CS plus. So our working hypothesis would be that the weights Wx and Wy are negatively correlated with cosine of theta CS and sine of theta CS, and negative sign is because both effects were pauses in the simplest case. So CS on is an unbiased estimator of the downstream potent axis of the Purkinje. So we extended the CS perturbation hypothesis by looking at different tunings of the complex spike. So some were more sharply tuned, some are less sharply tuned. So now the question is that how does it affect the downstream or not? And what we observe is that sharper the complex spike modulation, larger the Purkinje cell downstream effect on the eye movements. And here you can see on the right that the dark brown group is pushing the eye more than the light brown, which has the sharper tuning versus less sharp tuning. So not only the weights are depending on theta CS, but also depends on the rho, which is the amplitude of the tuning of the climbing. So as we discussed, this is a similar problem to motor cortex, but we have a priori knowledge about the weights mapping the neurons to behavior. So now the question is that, we're back to the question that the fact that these neurons' responses rates are bursting, pausing, they're lasting way longer than the movement duration, although their downstream effect supposedly is pulling the eye only toward a specific direction. So how can that be? And let's say that it's a null space holds in the cerebellar cortex. Let's assume that. And we have the benefit of having both W and N. So now we can answer the question that what is the downstream command M that is going, because we have both weights and the neural activity. But before that, first we're going to explore the linearity, the superposition, which means that do P cell responses add or cancel each other, which is technically if the W and if the model is valid or not. And second, we're going to check is if CS on is the potent axis and CS plus minus 90 is the null axis. In order to do that, because we have the simultaneous recordings, we have multiple simultaneous Purkinje cells. Let's say that the downstream effect of two simultaneous Purkinje cells is represented by rho 1 with angle theta 1 and rho 2 with angle theta 2. And let's just have vector sum of the two. And we're going to see that basically the net vector, the net prediction of the perturbation is going to depend on the ratio of rho 2 over rho 1 and the difference between the two angles. So we bin our pairs by the difference between their preferred direction. And as you can see, most of our cells have similar CS on tuning, have similar potent space tuning, but we have cells that have perpendicular tunings as well. So let's look at the perturbation. If two cells have the same preferred direction, their perturbation downstream adds with each other. And if it's perpendicular, you can see that the eye is pushed toward the second Purkinje cell perturbation. So if complex spike happens in both of these Purkinje cells, if they share the same direction in the pair scenario, the push is going to be more. If they're perpendicular to each other, it's going to be toward the vector sum of the two. So now that we showed this, let's form the population using the a priori weights that we found. So let's say that the ith cell downstream rate is Ri psi and T, which psi is the saccade direction. So this is the theta direction, this is the saccade direction, and this is in the foveal coordinate. So the Pi is expected to be the weights times the rate for this individual Purkinje, which we do a normalization term, but it would be basically minus rho I cosine and sine times Ri. So we expect the Purkinje cell with the potent axis to the right to only have a negative downstream effect on the horizontal burst generators and not to the vertical burst generators. Another information that we have is that actually the potent space has a spatial organization in the oculomotor vermis. With the right, the Purkinje cells located in the right vermis are pushing the eye to the right, and the Purkinje cells on the left pushing the eye to the left. So in order to remove that bias, we put isotropy assumption to remove sampling bias. And what it means is that we basically copy each Purkinje cell to 8 directions. I'm showing 4 of this here. That technically it means that I just rotate the Purkinje cell and rotate the weight. So this Purkinje cell has a sibling that is responding toward the opposite direction and has a positive downstream. There is one that is upward with similar responses to this Purkinje cell that has a negative downstream to the vertical burst generators, and there's an opposite to the down that has a positive response. And if you formulate this after a little bit of changes in variable, what you're going to see is that actually this population response does not depend independently to the saccade direction and the potent direction, but it depends on the side tilt, which is the difference between the saccade direction and the potent axis, which in fact, it means that you just need to do a rotation in your coordinate system to shift your coordinate system to the Purkinje cell coordinates instead of the foveal coordinate. And this would be psi minus theta. And for the purpose of simplifying the problem, we only consider the rightward saccades. And again, mathematically, we can show that because of the isotropy assumption, the population activity is going to be the same for all directions of the saccade. And the cosine and sine terms in turn would turn into projections on saccade direction and projection on saccade direction plus 90 degrees. And the population response is going to be the sum of all the p-tilde I pseudo populations that we have for all the neurons. So now let's test the hypothesis. So the hypothesis says that for a rightward movement, the Purkinje cells that have responses to the right and left are involved. So let's look at that response. As you can see, both of them have wider responses than the movement duration itself, both for bursters and pausers. But the theory predicts that a differential response of these two is what is controlling them. And if we do the subtraction, we're going to see a shorter response, a faster response with zero crossing with a change of sign at the deceleration onset. And more interestingly, for the pausers, we see the exact same response as we see for the bursters. So although bursters and pausers are behaving very differently, following the null space theory and following the weights that we found from the perturbation, the population response is going to be similar for the bursters and pausers. And also the prediction is that for the up and down response, for the rightward movement, the responses should cancel out each other and should go to the null space. And that is in fact what we see. There is a symmetry in the bursters response that cancel out each other for upward and downward movement, as can be seen here, and also for the pausers. And for diagonal responses, they are placed somewhere in between, as you expect. So in order to summarize, the first puzzle that we had, how are Purkinje cells involved in endpoint control despite the fact that they are active long after the movement ends? First, CS1 reveals the potent axis. Bursters and pausers show a similar projected response. And the Purkinje cell population pause is time-locked to the deceleration phase of saccade. So we can think of this phase, which I'm going to go through it in more details later on the slides, that this phase, we hypothesize that is stopping the eye, as you expect from the pause to push toward the opposite direction. In the perpendicular direction, there's no activity. So puzzle 2, can we generalize the potent space to other cell types? So as I went through the recordings earlier in the slides, I showed you that not only we have Purkinje cells, but also we have other cell types. Thanks to the work from Regular Lab, now we know that there are actually 2 subtypes of MLIs, and we have an idea about how the network is organized. So the theory is that the Mossy fiber projections are coming to the cerebellar cortex to the granule cells, and granule cells forming the parallel fibers. In the cortex, we have 2 types of MLIs, MLI2s suppressing MLI1s, and MLI1 suppressing Purkinje cells. And both MLIs and Purkinje cells receive parallel fiber inputs. And also from the work of Alt and Heuser, we know that one of the MLIs receive climbing fibers through a spillover, which we're going to show how we detect. So this is one of our recording. How are we able to tell apart the Purkinje cells from the MLIs and other cell types? So as I mentioned before, the Purkinje cells are easier to identify thanks to the complex spike suppressing them for tens of milliseconds and generating 2 types of spikes. But we use jitter-corrected cross-correlograms to identify the other cell types. So from the literature, we know that the simplest spikes synchronize with each other. So this is basically the probability of simplest spike given simplest spike corrected for the rates. So there's a millisecond synchrony in the simplest spike activity. MLI1s, we know that they have a phaptic coupling with each other and similar synchrony, but with wider responses. And we know that the MLI1s suppress simplest spikes within a millisecond, as can be seen here. And MLI2s suppress MLI1s within a millisecond. And in turn, MLI2s disinhibit the simplest spike. So this is how we identify MLI1s and MLI2s alongside the simplest spikes, in addition to the location information layer structure that we have here. Then we use a 3 millisecond causal window for these corrected cross-correlograms to form an adjacency matrix. And what we observe, in fact, is that not all the cells are connected with each other. Actually, there are cliques of cells, there are interconnected cliques of cells that are more connected to each other, but less connected to the rest of the cells, which is the adjacency, the layer information, the proximity in a layer is necessary, but it's not sufficient. So we do not put the cells necessarily into a clique because they're in the same layer. but the cells that are located in the same clique are coming from the same layer. So here I'm showing the between clique activities. So the red traces were within clique activities, and the blue are the between clique interactions. And as you can see, the between clique interactions are significantly less or not present at all. So to summarize here, Purkinje cells synchronize. You can see the block here. Red here, white means no, zero interaction. This is in hertz. Red means up to 20 hertz of synchrony, and minus 20 hertz blue means that there is suppression. The MLI synchronize with each other, and the MLI suppress the Purkinje cells. And in addition to that, we verified our MLI type identification by the fact that only MLI2s have an S spillover after climbing fibers happen, not the MLI1s. So now let's see what is the modulation. How do the MLIs respond during the saccade, and how can we interpret their population activity? So we hypothesize that the potent axis is shared within a clique. And the reasoning behind that is that we look at the multiple Purkinje cells that are located within a clique, and we check the CS1 direction angle difference between them. And we observe that the CS1 angle difference is significantly less within a clique than between a clique. So we align the MLIs. So through a clique, in a clique, we hypothesize that MLIs are Purkinje cells share the same potent direction. And we align the MLI1s and MLI2s to that potent direction. But the first observation is that the MLIs are actually bursting toward all directions of saccades with not that much directional tuning, which is surprising to us because we expected MLI2s to suppress MLI1s and MLI1s to suppress Purkinje cells, but none of these rates are showing opposite trends of activity. However, if we do the same projection as we did for the Purkinje cells, so here PXT is the component of the population activity toward the target direction, and PYT is toward perpendicular to the target direction. And as we discussed, for the Purkinje cells, for both bursters and pausers, we're going to see a burst-pause pattern, which the change in polarity happens exactly at the saccade deceleration, and we don't see much activity in the perpendicular directions. For the MLI1s, interestingly, we see an opposite pattern to what we see for the Purkinje cells, supporting the idea of the MLI1 suppressing the Purkinje cells, and also can be seen in the phase plots of the Purkinje cells rate as a function of the MLI1 rates. And for MLI2s, the population activity is opposite of the MLI1s, as we expect. And again, a phase plot of the MLI2 as a function of MLI1 displays the negative interaction between the MLI2s and MLI1s. So we believe that putting the MLIs together using the shared potent vector in a clique would reveal the population response of the MLI. So interneurons inherit the vector space of the Purkinje cells. So now we ask, so we have these population responses in time, but what do they mean? What is being computed by the network? In order to do that, we're going to look at a little bit of the studies that have been done about the control of the saccade. And as we know, the superior cliqueless, using the information that is coming from the cortex, drives the saccades. And the stimulation of the superior cliqueless initiates saccades. A copy of this command, a copy of this sensory goal, through the NRTP, nucleus reticularis tegmenti ponti, goes to the cerebellum through the Mossy fibers. And superior cliqueless has projections to the burst generators that in turn derive the ocular motor neurons. And we know that from the studies from Noda, Peter Thier lab, and other people, that also the burst generators send copies through the Mossy fibers to the cerebellar cortex. So we're building ideas of Lance Optiken work that in order for the cerebellum to be able to control the saccade, to land on the correct target, you first need the sensory goal, where I want to go. And second, you need a real-time readout, that's where I am, like a forward model. So which a copy of burst generators will tell you that state information, and a copy from the NRTP can tell you that sensory. So I first start with how we identify the Mossy fibers. as traditionally has been done, people have used the layer information and the granular layer. They use the waveform of the Mossy fibers to identify them. As you can see, the Golgi cells have wider responses, and the Mossy fibers have these M shapes with negative afterwave or sharp waveforms. And this has been shown in multiple species and multiple papers. And what we did here was that we used NeuroPixel's ultra recordings, which are these high density NeuroPixel probes, to basically show that a single Rosetta structure, a single Mossy fiber, has both waveforms based on the location of the recording. So here you see the M shape with negative afterwave, and here you see the fastest spiking that transitions over the space to each other. And we used UMAP in a spatial temporal waveforms, which is basically we have multi-contact probes, so we can have the distance from the center of the cell, whatever recording site it is, and we can present it in time. So we use these images to classify the neurons that we had in the granular layer to identify the Mossy fibers versus the other granular layer interneurons. And obviously, as we expected, we had a group that had the M shape with negative afterwave. We had fastest spiking, which we believe both these clusters that are represented here are belonging to the Mossy fiber group. And we have another group that is way larger in a space, and also in time, it has a wider spike. So if we look at the activity of these Mossy fibers, I'm going to show you one example of state Mossy fibers activity to the targeted saccades. And here each raster is presenting movement direction. So this is upward direction, this is downward direction, left and right. And as you can see, this cell prefers the downward direction. And if you look at the profiles, and the saccades are sorted by the saccade duration. So you can see that the burst duration increases as the movement duration increases. And the responses, the rates are really resembling the velocity profiles. In addition to that, as shown from the burst generators, these cells are really pushing the eye toward the direction. So each individual spike can be regarded as pushing the eye a little bit toward the preferred direction. So here I'm showing that the amplitude has a linear relationship with number of spikes. Not only for the targeted saccades, but also for the spontaneous saccades. So this is literally just kinematic dependent. So if you want the eye to be moved, these neurons are going to be active with the same properties. The only difference is that these movements are just slower, so the burst durations are going to be longer. But the same slopes for the number of spikes versus amplitude are being observed on the same rates. However, we have a second type. Sorry, this is a typo. This should be example gold Mossy fibers. We have another type that the responses are different and do not seem to be solely kinematic dependent. Here is an example gold Mossy fiber, and you can see its preferred direction is actually diagonal, it's to the left up direction, and it has a movement field, meaning that it's responding for a specific location, for saccades toward a specific location in the space, not larger and smaller. In addition to that, the responses are way less for the controlled spontaneous saccades that are out of the context of the task. These Mossy fibers have been shown previously in the field too. So this is the study of Otsuka and Noda, 1992, and they showed that many of their Mossy fibers have directional tuning and amplitude tuning. The work of Peter Tier lab has shown many of the burst generator Mossy fibers that are basically linearly, the peak discharges are related to the amplitude, and also the saccade durations being related to the burst offset. So we use this information to classify a subset of our Mossy fibers to state Mossy fibers and gold Mossy fibers. And what we observed was that actually our state Mossy fibers were in the motor coordinates. Their preferred directions were left and right and up and down, while our gold Mossy fibers' preferred directions were in the sensory coordinates, as in colliculus, that we could have had diagonal preferred directions. In addition to that, as we discussed, this is just a population of the single cell that I showed. The state Mossy fibers, their number of spikes is linearly related to the saccade amplitude, no matter if you have reward relevant or reward irrelevant movements. But the gold Mossy fibers have usually movement fields, and they differentiate, their responses are different for reward relevant versus reward irrelevant saccades. Okay, so back to our main puzzle. What is the population computing? So let's start with the targeted saccades, and let's come up with a model. So We have the sensory goal, visual space through the goal Mossy fibers, XG, and we have the motor command, the muscle space, through the state Mossy fibers, U. And let's say that if we have an integrator, we would be able to have the displacement, as we discussed. And the simple boundary would be able to tell when the Purkinje cell need to pause to stop the movement. So what we observe in the responses, we bin the Purkinje cells and plotting the Purkinje cell responses in the projected space as a population, and MLI1s one step before. MLI1s, just as a reminder, we're suppressing the Purkinje cells population responses bin by the velocity. So what we observe is that the Purkinje cell is still crossing zero at deceleration onset, and the MLI is reaching a peak here, and the manifold is still negative. A phase plot of the Purkinje cell as a function of the MLI. We observe that for all the bins of the saccade velocity, the zero crossing is there, and MLI is reaching the same peak, and also the MLI1 and Purkinje cells remain on similar manifolds. However, what happens if you look at the spontaneous saccades, that we don't have the sensory goal information, but still we have the state information. So what we observe is that the MLI1s and the Purkinje cells are still responding for similar peak velocities, but the MLI1s are not anymore reaching the same peak as they used to reach. And the Purkinje cells are not crossing the zero, are not stopping the eye anymore. However, the two responses are remaining on the same manifold. But this time, the zero crossing, as here identified the deceleration onset, here does not identify the phase of the deceleration in the movement. So the lack of the sensory goal disable the Purkinje cells to be able to estimate when to stop the saccade. So in order to further explore this effect, we bin the saccade these times by their acceleration duration. And why acceleration duration? Because saccade starts and the deceleration is going to happen sometime in future, and this duration is acceleration duration. So for short acceleration duration, we see the zero crossing in the Purkinje cells, but almost for double this duration, although the initiation of the activity of the population of the Purkinje cells starts earlier, but the zero crossing still happens at the deceleration onset. And the responses are negative here. And here we are just quantifying the zero crossing of the Purkinje cell that remains around zero, no matter what the acceleration duration is. And the MLIs are reaching the same peak, no matter what, around 40 Hertz for targeted saccades for all bins of the acceleration duration. And the MLI1s and Purkinje cell remain on similar manifold still, with zero crossing of the Purkinje cell identifying the deceleration onset of the saccade. However, for spontaneous saccades, for other saccades, the MLI1s are not reaching the same peak. It's almost half. And the Purkinje cell zero crossings are not happening. So Purkinje cell signal when the saccade should begin decelerating, but only if the Mossy fibers provide location of target, the sensory goal of the movement. So in order to summarize, we try to tackle three puzzles. Puzzle one was how are P cells involved in endpoint control despite the fact that they are active long after the movement ends? Specifically for bursters and pausers that seem to have very different modulations to the similar movements. And the answer was that inferring the weights from a perturbation analysis through the climbing fibers was able to give us a population response that is the same for the bursters and pausers, and is only active toward the movement direction. Our puzzle two was that if we can generalize the potent space to other cell types, and we formed the cliques of interconnected MLIs and Purkinje cells, and we were able to expand the idea of the potent space, the weights, to the MLI1s and MLI2s, which revealed the functional connectivity between these cell types, which was expected from the anatomy in the phase plots that you can see here. And the puzzle three, which was what is being computed by the cerebellar cortex. And we believe comparing the spontaneous saccades versus the reward relevant saccades, the population activity is indicating the deceleration onset only when the sensory goal is information is brought to the cerebellar cortex through the Mossy fibers. So I want to thank all Shadmehr lab, specifically Reza, for giving me the opportunity and also his fantastic mentorship. Alden has been of help for setting up the whole electrophysiology lab and the silicon probes and recording in the animals. Paul as well. And Hisham has helped with the perturbation analysis with the complex spikes. Solomon has been always kind to help with the theoretical aspects and just thinking and being always available, which is fantastic. He's a fantastic thinker. Jinu was the master's student in our lab, which helped with saccade detection and developing the UNI for the marmosets. And Jay has helped with the software, the behavioral software that we use for training of the animals and record. And obviously our monkeys have been the real heroes here. Thank you all for your attention. And I'm glad to hear from you.
Wonderful. So yeah, it's open for questions, and please just go ahead and raise your hand, and we can listen to your comments and get feedback.
I can't figure out how to raise my hand, but I can talk.
Wonderful,
Can
David.
I
Go
ask
ahead.
a few questions?
Of course.
Okay, this is fascinating as always for me because I'm looking and seeing potential clinical relationships. I want to talk about pushing of the eye, and in other words, biasing of the eye. And a couple points about that. From the clinic, we know in Wallenberg syndrome, which I know you know about, that when the patients have an asymmetrical lesion, which presumably is affecting the way the Purkinje cells on one side of the vermis are functioning, and in turn their effects on the vesicular nucleus, you get a bias. If the animal tries to make a horizontal, or the human, excuse me, although you see the same thing if you put some neurotoxins in one vesicular nucleus in an animal, or if you make an asymmetrical vermis lesion. Anyway, you make a horizontal saccade, it's big in one direction and small in the other. There's no rotation, there's no inappropriate vertical component, but when you attempt to make a vertical saccade, you get a bias such that the eye doesn't go straight up or down, but it's biased in the same direction the horizontal component was when you made a horizontal saccade. So it's not just a rotation of the entire vector space, if you will, but that there's a horizontal bias that's added. So my point is, would your interpretation and your results be compatible with these kind of, and explain perhaps these peculiarities of the dysmetria that's created when you interrupt, and this is the presumed mechanism, the climbing fiber input?
This is a great question. Thank you, Dr. Z. So actually, I agree with you. So the interesting part is that these Purkinje cells, like as you mentioned, so if I understood correctly, correct me if I'm not understanding your question correctly, but these Purkinje cells, although they're caring about left and right, they're active for up and down movements. But in a regular saccade, in a context of no like a special learning or any disease, basically the up and down response are canceling each other. So any disease or any induction of learning that perturbs that breaks that symmetry is going to push the eye toward that direction. So for upward saccade, I expect it to be pushed to the right because the competition between the left and right vermis is not going to hold anymore because one side is going to be less active. So the responses for the upward is not going to cancel out each other between the left and right vermis.
And the response for the downward saccade, what would that show?
There's actually the symmetry would in turn would be that up and down for each individual cell is similar. But our idea is that because there's an isotropy, so basically what we think is that the Purkinje cell that is sitting on the right vermis has a sibling in the left vermis. that its responses are just rotated of that. So in our individual Purkinje cells, what we see is that usually their up and down responses are similar. There's a symmetry. And their left and right responses are dissimilar. So
See,
what
go ahead.
happens, So what happens is that when you make an upward movement for the Purkinje cell that is caring about the right, the Purkinje cell that is sitting on the left vermis is going to have a response for the up as the down response of this Purkinje cell. So that up and down cancel each other, as illustrated here. I mean, let me go to actually this slide that I have it separated better. Excuse me. There we go. Okay, so here when I identify null, so this is the response of the technically right Purkinje cell for the, let's say, up. This is the left Purkinje cell for the up. And they cancel out each other. So if there's any perturbation in one of them, this push-pull system would not work as well as it's supposed to work. So the cancellation doesn't happen. And the downstream of this Purkinje cell is pushing toward the right. So if there is a mismatch, the push is going to be to the right, although you're making an upward saccade, as the simulation studies also showed.
So if you make my question is, if you make an upward saccade versus a downward saccade, is the effect the same on the horizontal component? If you took out one vermis on one half side and you looked up and down, would you see a horizontal bias with those vertical saccades that would be in the same direction, or would they be in the opposite directions, up versus down?
Same direction. So all the pushes, no matter what the initial saccade is going to be toward the same direction.
Oh, okay, that's good. That makes sense.
Yes.
On a related, very technical point, in humans, when you look up and down, there's a clear asymmetry. When you look up, the two eyes tend to go apart, a little bit out, and when we look down, the two eyes tend to go together. We call that a Venn pattern, looking up and looking down. It kind of makes sense, because when you look down, you usually want to converge your eyes. When you look up, you want to look far apart. So my question is, if you're looking at these effects of the Purkinje cells during random saccades in many directions, are you recording from one eye or both eyes? Are the eyes converging? Or and if you do both eyes, are they doing the same thing in terms of the Purkinje cells?
Yeah, this is again a fantastic question. So right now I'm only recording from one. We have the recordings for both eyes, but I'm only analyzing one eye. And also there is evidence in the literature that the Purkinje cells are able to control each individual eye separately, which is actually going to be interesting for the saccades that have the depth component, because the amount of movement for each eye is going to be different. And as you mentioned, if there are any differences in the up and down movements, but right now we are not considering that in this analysis.
Okay.
Solomon, go ahead. You have your hand up.
Thank you.
Thank you.
That's fantastic. Can you try to do something similar with the Mossy fibers? So try to kind of guess the weights of the Mossy fibers. I know you have to skip. I'm sorry. I know you have to skip the granule cells. But what happens, if you take all the Mossy fibers within a region and you just kind of took the average of them, do you see something similar to what you see in the Purkinje cells? Or do you have to start guessing their weights through the granule cell MLI system?
So the distribution, so Mossy fibers are not necessary. So that is again a great question. So if I understood correctly, Mossy fibers, unlike the Purkinje cells, their preferred direction is toward all directions. It's not like the CS on that in this location I have a specific CS on. So here, as I showed here, the information about all directions is almost present.
Okay.
And you expect that.
Yeah. So the idea you would have to add some modulation that is happening by the complex spikes. But the idea is that the fundamental shape of the simple spikes is determined just by some averaging of Mossy fibers.
Oh, I see what you're saying. Yeah.
So yeah, I agree. The difference between the CS on and off will have to be determined by the CS, but the actual response can be some averaging. My point is, can we try to guesstimate the weights of the Mossy fibers, even though we're missing another layer?
Yeah, like fitting a dynamical system.
Yeah.
And I tried that. And we can discuss it further probably offline. But the point about the state Mossy fibers is that there's a delay component too, which we don't know why it exists. With the goal Mossy fibers, they're usually very time locked to each other, but the state Mossy fibers have a delay response, which people usually think about it as in the granule cell happening. But our Mossy fibers already have a delay representation of response. So, and that seems to be fundamental because of many of these Purkinje cell responses and MLI responses are double peaks, and how this population, one during the acceleration, one during the deceleration, and how these population responses are formed, many of them is the lack of the second peak in the deceleration or more pronounced presentation of in the deceleration. So, yeah, I think it's possible, but it requires more thinking, and obviously I'm open to it. If you want, we can like brainstorm more about it.
Okay, thanks.
Sam,
Thank you.
Sam, please go ahead, Sam.
Hey, Mohammed, that was a wonderful, rich talk. Really impressive work. I had a question, I think related to the previous one, maybe. I might have missed something. I was trying to think about the temporal dynamics of the state information coming in to your population, because I guess you framed it as as contributing to maybe a forward model type computation. And if that's so, you'd want it to be preceding the movement of the plant, right? So I'm just curious if there's some analysis you could do there to get that sort of causal arrow of time sort of issue worked out.
Yeah, definitely. Let me see if I have there is one slide that I'm missing here that maybe I can open it like really quickly for you. So the Mossy fiber responses, oh, not this one, sorry. So here, I can zoom in. So as you can you see my screen, or okay, now it should be better. Okay. Yeah, so the state Mossy fibers.
Desktop right now. I'm just seeing your desktop.
Okay. Yeah. Sorry. Okay, so
there we go.
Here. Okay, good. Okay, good. So the state Mossy fibers here, I'm plotting the response aligned to time from deceleration, and each row is a Mossy fiber. And as I mentioned, so it's very interesting, and I honestly don't know what is the purpose here that these guys, specifically the late guys, are serving, because many of these late guys are way after the movement ends. And we have early ones, and the early ones are actually earlier than the goal Mossy fibers, as you expect, because these are closer to the muscle. If they're really the burst generators, if they're actually from the EB and IBNs, as the anatomy suggests, they're going to be closer to the movement itself than a copy of colliculus going through an RTP.
Cool.
So,
That's really interesting.
so the integration, we don't have any evidence for that. So we were hoping that maybe we can find some cells in the granular layer, like UBCs, or even the Golgi cells themselves, that are doing this displacement integration. But I mean, I haven't looked at the data probably as much as I'm supposed to, but just looking at their modulations, we have neural integrators, which are actually position encoding. They're position integrators. They're not displacement integrators. And they're coming through the Mossy fiber inputs. But we haven't found the cell type that specifically is doing the displacement integrator, as the model suggests. So our current working hypothesis is probably the MLI1s might have been involved in this, because no matter what the peak velocity or saccade amplitude is, the MLI1s reach the same peak around the time of the deceleration. So this might be the integration that is happening. Also, it's not present when you don't have the sensory goal. So what is the purpose of integrating when I don't know where I should end the integration? So yeah, I don't know, but we are not sure about the biological substrate of this displacement integrate.
Thanks.
Gabe, you have your hand up, please.
Hey, I mean, really great
Hi,
stuff.
Gabe.
Hey.
Yeah, thank you.
So I'm trying to better understand how you're thinking about the goal Mossy fibers and potentially originating from the collicula. So my question is, what do you think the P cells would do if you generated a saccade by stimulating the colliculus?
Yeah, that's also a great question. I don't know. I don't know. Like the hard part is that actually these Mossy fibers that are coming through the NRTP are not necessarily colliculus, because most of them do not have the visual component. So NRTP, as in colliculus, has visual neurons, visual motor neurons, and motor neurons. And what we see as this in this Mossy fibers is that they mainly motor. Very few of them are visual motor. Maybe one or two out of like 1,000 that we have is visual only. So it's a selection of the cells that are like it's pruned. It is not a representing sample, unbiased sample of the colliculus. In addition to that, the work of Ziad Hafed, like colliculus is like very selective to different visual stimuli and different types of movements. So, and again, it seems like that the ones that we see that are being projected to the cerebellum are the ones, at least for the context of our task, are the ones that are not for the spontaneous saccades, but are for the targeted saccades. But obviously, I think there are evidence that there are both types of cells. I think Ziad Hafed has a paper that colliculus neurons, some of these colliculus neurons are more responsive to memory-guided saccades than the visually guided saccades for the similar kinematics. So given that heterogeneity in the colliculus, and at least given this set of data set that currently I have, I'm not sure.
Well, I guess my next question was going to be, how about for memory guided saccades? Like, is it really that it's goal directed, or is it more so that there's a visual target that the animal is saccading towards?
Yeah, these are fantastic questions. And you have an internal goal, obviously, for a memory guided saccade. And I think what is more important here, at least what I mean, we should test this based on this data. I don't think I'm able to answer your question. But I think what would be interesting is that here, I think what matters is that it's vital for me to make accurate saccade to get reward. So I care. So the information should go to the cerebellum to stop the collicular activity, to not be inaccurate, to land on the target accurately. I think that is what is going on. I'm hoping that we're going to have more answers with our postdoc tasks, that he's like looking at now having different values for these visual stimuli and making the saccades, and see how these Mossy fibers have different responses and how the Purkinje cells have different responses to them.
Thanks.
But I can tell you from our behavioral studies, if there are different values, monkey gets sloppier, like their saccades get less accurate.
Yeah, really interesting. Thanks.
Thank you, Gabe.
Thank you.
If I can just say one thing, I think that's such an important point. I don't think there ever is a saccade without a goal.
Definitely.
And the vestibular system would be a classic example, but maybe it's how accurate that saccade has to be, which is why the cerebellum gets involved. So you really need to test that hypothesis. It's a real important point.
Definitely, yeah.
Any other questions? Well, Happy New Year, everyone. Good to see you. Thank you so much.